All right, so welcome to the second video for our animal module. All right, so the first video we talked about evolutionary history of animals, what we thought the the common ancestor to all animals are, and the Cambrian explosion. So when we see the vast array of body plans really start to come about during the Cambrian period. So now in this video, we're going to go over the evolutionary history of animals and look at the phylogeny, the phylogenetic history of the major phyla for the animal kingdom. And then we're going to start off by looking at the first two groups of the animal phyla, the sponges and the Cnidarians. Okay. And so in first thinking about how animals are related to one another and how we should group and categorize animals and put them in their evolutionary trees. Early on, biologists categorized animals based off of their body plan. So basically the general features that we see in animal body structures. And the distinctions between the body plans were used to generate the phylogenetic trees which led to the development of the evolutionary relationships that we looked at between animals and animal groups. With the development of genetic data and the ability to sequence the genomes of animals a lot more efficiently, it's allowed us to basically refine our evolutionary trees and relationships of animals that were based on those body plans. And so now we have these evolutionary histories that go off of body plans and off of genetic data, right? So we can kind of refine those older trees with our newer data. And early on, there was this major branching point in the evolutionary history of animals that starts off with the most basal of the animal phyla, and that is the sponges. And the sponges are different from all of the other animals because they kind of really lack the structural complexity that we see in all of the other animal groups and animal phyla. Sponges don't have a true formed body. It doesn't have a real true body plan, so they're asymmetrical. They don't have true tissues, and they're basically made up of a group of cells that all perform the same function. So really similar to what we think the ancestral animals look like. These cells that have a flagellum, they have these collar cells that filter the water, and they're all working together in this one structure that is kind of serving the same purpose. And so that's why we put sponges on the basal portion of the ancestral or the evolutionary tree of animals because they don't have any of those complex features, and they really resemble what we think the ancestral protist early animals would have looked like. And if we look at this evolutionary tree here, think about which animals are more closely related to chordates in this evolutionary relationship. Okay. And so just pause the video, take a minute, and answer that question. All right, so hopefully you got this based off of our previous discussion in the evolution chapter and said that echinoderms are more closely related to chordates than any of the other animal groups. And if you did, then you would have been correct. So chordates and echinoderms are sister phyla, okay? And so that basically means that chordates and echinoderms are more closely related to each other than they are to any of the other groupings like arthropods, roundworms, annelids, flatworms, etc. Okay. Now, with this phylogenetic tree, there are a couple of things going on here that I want to talk about and mention. And most of these are shown in these blue boxes here. So these are key traits and characteristics that I want to discuss and when they arose and what groups they're present in terms of the evolutionary history of animals. Okay, so we said that the animal groupings, the animal phyla, are separated based off of similar body plans. Okay, and so this is what these blue boxes are kind of getting at. So the first big separation is whether or not animals have tissues or no tissues. Okay, and what we can see is that the ancestral protists likely did not have tissues, and tissues evolved after sponges evolved, right? So you see sponges have no tissues, but this here is saying that everything after sponges have to have true tissues. Okay. Now, Cnidarians, they only have two tissues. Okay, they only have two sets of tissue layers. So I'm going to make some more markings on here. So we're going to say two tissues. Sorry for the sloppiness. Using my mouse pad on my laptop doesn't really work well for writing. Okay, so Cnidarians have two tissue layers. Everything after Cnidarians has three tissue layers. So I'm just going to mark that here with a 3. Okay, so 3 tissue layers. Okay, so that's a big difference between the tissues. So sponges, no tissues. Cnidarians have 2 tissue layers, and everything else has 3 tissue layers. Now, not only is tissues important for separating organisms into phyla, but also symmetry and how the body is laid out. So sponges are asymmetrical, right? So no symmetry. So no symmetry. And I'm just going to do symmetry as SYM. Okay, so no symmetry. Cnidarians are radial, they have radial symmetry, right? And we'll talk exactly what that means in a second, but basically you can cut a Cnidarian on any plane and it will be symmetrical. All of the other phyla are bilaterally symmetrical, which means you only can cut them in one plane and they're symmetrical only on that plane. If you cut them on any other plane or separate their bodies on any other plane, they won't be symmetrical. Okay. The only phylum that is not bilaterally symmetrical is echinoderms. And I'm going to clarify this statement in a second. And so echinoderms, they have radial symmetry. Okay, so I'm going to say rad for radial. Now, this is secondarily radial. So they basically re-evolved radial symmetry after it was lost in the other phylum. But what's interesting with echinoderms is their larvae are bilaterally symmetrical. Okay, their larvae are bilaterally symmetrical, and the adult forms are radially symmetrical. Right? That's a little interesting thing about echinoderms. Now, there are some other ways we group these animals as well. So we have what is known as Lophotrochozoans and ecdysozoans. Okay, so here would be your Lophotrochozoans. Okay, so that's LOP. It's a long word. I'm not going to write it out, but I'll show you in a little bit what it is. That's Lophotrochozoans and ecdysozoans. We're just going to say EC for ecdysis. And what this basically means is that these phylum, animals in these phylum, increase their body size by increasing their mass. Okay, they just grow in their body size. Ecdysozoans, they increase their body size through molting. All right, so notice arthropods are here. So they have exoskeletons, and so they molt those exoskeletons. Okay, so those are two differences. Now, a final difference that really separates phyla on this tree are the way their early development happens as an embryo. And we can have what's called protostome and deuterostomes. Okay? These organisms here are protostomes. So we're just going to say PR for protostomes and then DE for deuterostomes. Okay, so echinoderms and chordates are deuterostomes, and all of the other bilaterally symmetrical phyla are protostomes. Okay, and so basically so we'll get into the difference between protostomes and deuterostomes here in a second, but the key thing right now is just knowing that there are some differences in that early development that happens. Okay, so with this phylogenetic tree, it's actually telling you a lot if you map these characters on here. So by looking at this final tree that we've got annotated, we know that sponges don't have any symmetry. They're asymmetrical. They have no true tissues. They're the most basal group of the animal kingdom, and they differ from all of the other phyla in that they don't have any tissues, right? Now, all of the other phyla have tissues. However, Cnidarians, which is the second most basal on this tree that we're showing here, only has 2 true tissue layers, whereas everything else has 3 true tissue layers. Cnidarians also are radial. They have radial symmetry, whereas everything else is bilaterally symmetrical. Okay. Now we can further divide these phyla that are bilaterally symmetrical into protostomes and deuterostomes, which have differences in their embryonic development. We'll talk about those in a second. Protostomes can be further divided into Lophotrochozoans and ecdysozoans, based off of whether or not they molt during their growth and development. And then finally, your deuterostomes is separated into echinoderms and chordates. Chordates are your vertebrates, okay, so like your mammals. And echinoderms have that secondarily radial body form, right? So the larvae are bilaterally symmetrical, but the adults have that radial symmetry. Okay? So these are the big characteristics that you want to know for each of the phylum, and we're going to go and talk about these in more detail as we continue through our module. And now I mentioned I was going to talk about these protostomes and deuterostome features and what they actually mean, and we're going to do that now. Okay, so whenever you have fertilization, right, that zygote is formed, and it will start to undergo mitosis, and it will start to divide and make more cells as that zygote gets bigger. that forms a blastula, which is what we see here on the left-hand side of these figures. So you have a blastula. Now, the cells on this blastula will invaginate and lead to what's called a blasteopore. Okay? This blasteopore is an opening in the organism's digestive tract. Okay? One way to know whether or not the animal is a protostome or a deuterostome is the final function of this initial opening. In protostomes, this opening forms the mouth, like you see here. In deuterostomes, that opening forms the anus, like we see here. And what you also see is that different tissue layers are developing as well. So you have your ectoderm, which is like your skin, your endoderm, and your mesoderm. Right? We'll talk a little bit about that in a few minutes. But basically, whatever the blasteopore opening develops into tells you which of these groups it falls in. So if the blasteopore forms the mouth, then it's the protostome. And if it forms the anus, it's the deuterostome. But eventually it will lead to the formation of a complete digestive tract where it will have two openings, both a mouth and an anus. Okay. And notice we're saying a complete digestive tract because not all the organisms shown here have a complete digestive tract, right? And so that's another important aspect to kind of pay attention to as we talk about each of these different phylum. All right, so we talked about how the animals can be split in terms of radial symmetry and bilateral symmetry, and what exactly those mean. Right, so radial split on all planes. Bilateral is only split in one plane where the two halves are the same. Now, another thing that helps to distinguish organisms is the development of a body cavity. So a body cavity is what we just refer to as a fluid-filled space that separates the digestive tract from the outer body wall. Okay? And I think I have an image here where we'll show exactly what a body cavity is. First, we're going to look at symmetry. So these are examples of radial symmetry, right? So you can, anywhere you kind of slice this anemone is going to be symmetrical. Any kind of plane you slice this pot is going to be symmetrical. However, with bilateral symmetry, it's only one plane, usually from head to tail on animals, and then both of those sides, the resulting sides, will be symmetrical. So that's the bilateral symmetry. For body plans, there are three different types of body plans. You have eucoelomates, acoelomates, and pseudocoelomates. Basically, these body plans exist in animals that have three tissue types, so your bilateral animals. You notice here there's three different colors. These three colors are associated with a tissue layer. The blue is the ectoderm, so it's like your skin. The yellow is the endoderm, and that is your digestive tract. The red is the mesoderm, and the mesoderm can vary depending on the organism that it's in. So, for example, a flatworm has no body cavity, so those mesoderm cells completely fill in the space between the digestive tract and the outer tissue layer. So there's no cavity, there's no space opening in that organism. And that's referred to as an acoelomate. Now, an earthworm and humans have a true body cavity. So this mesoderm doesn't fill in the entire space in between the endoderm and the ectoderm. It lines the inside of the ectoderm and it lines the outside of the mesoderm and kind of connects those two layers together with some space in between, and that's the body cavity. That space is the body cavity. Okay. And you can think of this as like the connective tissues that are keeping the organs suspended, the digestive tract suspended and in place in the body. Okay. Now, this is called a eucoelomate. Okay. We also have a pseudocoelomate, and I don't have an image here, but I could draw one real quick. So what a pseudocoelomate is, basically think of a eucoelomate. So we have the ectoderm. That yellow is hard to see. We have our endoderm, and then the mesoderm is still there, but it only lines the inside of the ectoderm. It does not line the outside of the endoderm. And so there's not a true body cavity here, right? There's not this true cavity that's formed by those two layers. Okay, so that's the difference between a this is bad, an acoelomate, and then a eucoelomate is just an EU coelomate, and then a pseudocoelomate, which is this one I drew, is pseudocoelomate. Okay. Now, looking at the first two phylum of the animal evolutionary tree, right? So all of these characteristics that we just talked about here are how we divide animals into their different groupings, right? So it's based off of body plans, tissue layers, developmental processes, all of those factors, right? Body cavity shapes, those sorts of factors. Okay, but now looking at the first two phylum of animals, these are invertebrates, and if you actually look, animals are represented largely by invertebrates. All of these organisms here are invertebrates. Your vertebrates are here. So 95% of the animal kingdom is made up of animals without skeletons, without backbones. Sponges are the most basal phylum of animals, and their phylum name is Porifera. Okay, so you'll want to remember these phylum names. And Porifera, basically pores, sponges have lots of pores, is the sponge phylum. So this is where all of your sponges are located on the evolutionary tree. So these are stationary animals. Like we said, they don't have true tissues, and we think they evolved from colonial protists. And we'll see like the structure of sponges and how they really resemble those colonial hypothetical ancestors. And so basically a sponge, you can just kind of think of a sack of cells that have a lot of holes in that sack, okay? And the sack is made up of choanocyte cells. Man, that's bad. It's so hard to draw with this thing. So choanocyte cells, okay? And these cells have a flagellum that will beat up and down, back and forth, that causes a water flow throughout the sponge. Okay, so we can see here that you have these choanocytes. So this single cell is a choanocyte. And these choanocytes are embedded in a matrix. And this matrix is made up of substances and spicules. And these spicules can be made out of calcium, they can be made out of silica and other structures, and that leads to different types of sponges. So if it's a silica spicule, then it's a glass sponge. If it's a calcium spicule, then it's like a harder sponge. Whereas the softer sponges will be made out of a different type of spicule, like a sponge like you can imagine what you would like wash your dishes with, right? Those are synthetically made sponges, but before we made those sponges, people would literally use like a sponge to do that type of stuff with. Um, and with these choanocytes, notice that there is flagellum that are on the end of these choanocytes. And that flagellum, again, beats back and forth, and it pulls water through the pores from outside of the sponge to the inside of the sponge into that central cavity. And then that water will flow up and out of that opening in the top, like we see here. And as that water flows across that choanocyte, it's filtered by a collar cell. And that collar cell is that little kind of collar that's at the end of the cell. And basically, what happens is it will trap organic molecules. That organic molecule will move through the collar cell to these amoebocytes, which are these blue structures here. And then these amoebocytes will move the food around the sponge, and then that food will be broken down and turned into energy that will allow the sponge to grow and develop. And so you can see how these colonies of choanocytes, these cells, are working together to help keep that sponge active. All right, so the next phylum is the phylum Cnidaria. So this contains the organisms that we refer to as Cnidarians. Cnidarians are like your sea anemones, your jellyfish, etc. Cnidarians are the first animals that have body tissues. However, they only have two tissue layers. They have an ectoderm and an endoderm. They do not have a mesoderm, so they don't have the red tissue layers that we were just looking at. They only have the outer tissue layer and the digestive tissue layer. So their basic body plan is just a sac with a central digestive cavity, so this gastrovascular cavity. Cnidarians have radial symmetry, and then they're characterized by these special stinging cells that are called cnidocytes. And that's where the phylum name comes from. All cnidarians have typically two variations in their body plan. They'll have a polyp stage, which is stationary, and then they'll have a floating medusa stage. So a sea anemone that is attached to a rock that has like the little tentacles is a polyp. A floating medusa is like you think of a jellyfish kind of floating in the water, right? So these forms here. And so you can see you only have the two tissue layers, the outer tissue and the inner tissue in both of these cases. They only have one opening, so they don't have a complete digestive tract. So the opening serves as both their mouth and their anus. They don't have separate openings for that, so it's not a complete digestive tract. It's an incomplete digestive tract. And so again, you have species like sea anemone, hydra, corals, all your jellyfishes. Those are your types of cnidarians. And so even though a jellyfish is predominantly a medusa stage, its reproductive stages are polyps. And so the polyps will actually release the medusa stage, and the larvae will grow into a polyp that will then again reproduce and release the jellyfish medusa stage. And so all those organisms have either that polyp or medusa form. Now, some species only have the polyp form, right? So little differences there. Now, cnidarians are carnivorous organisms. They use their tentacles with their cnidocytes to grab onto the food and basically push them into their opening, their digestive opening, and then they'll digest their prey that way. And like we mentioned, the tentacles are contained, they contain these cnidocytes, which again is where the phylum name comes from. Cnidocytes are basically just stinging cells, and they either can help with defense or they can be used to paralyze and help capture prey. So this is what a cnidocyte stinging cell basically looks like. So you have a hydra here. We're going to zoom in to the tip of one of their tentacles. And you see there are these cells here that have a little hair kind of above the skin layer. That little hair-like structure is a trigger. So as soon as something brushes against that trigger, this stuff on the inside, the coiled thread, will be released. And then it will come into contact with the prey, and that is what releases the toxins and the stinging chemicals that either paralyzes or subdues the prey, or serves as a defense mechanism. Okay? So that's basically how those cnidocytes work. Okay. All right, and so that's it for kind of our most basal early groupings of the animal kingdom. What we're going to look at next are the Lophotrochozoans. Then we'll talk about the Ecdysozoans, and then we'll go into the Deuterostomes. So we're going to break the protostomes up into two different videos, and then we'll break the Deuterostome into its own video. Okay, so we still got 3 more videos left. Like I said, a dense module, but we'll get through it slow and steady. And so we'll see you in the next video.